Okay, so Bache, this gets us to one of our biggest topics in general microbiology, as well as one of the most important ones, that is sterilization and disinfection. सबसे पहले, before I even go into the nitty-gritties of this chapter, what do we mean by sterilization? इसका मतलब क्या है? What does sterilization mean? Sterilization, this is a process by which a surface is made free of all the organisms and spores. वो वाला surface ना sterile हो गया है. Sterilization. This is a process. This is a process by which. This is a process. This is a process by which a surface, by which an article, by which an article or surface is made free, by which an article or surface is made free of all microorganisms. It is made free of all microorganisms, either vegetative state, either in the vegetative state, either vegetative state or spores, or spores. So point to be noted here is that the surface is totally free of all microorganisms. The surface is totally free of all microorganisms, including spores. That is sterilization. The surface is totally sterile. Versus disinfection. Versus disinfection, which is associated with destruction of all pathogens. Disinfection, which is associated with destruction of all pathogens, but not spores. It is associated with destruction of all pathogens, but not spores. Disinfected है. Spores are as such formed in unfavorable conditions. So in disinfection, the area is disinfected. It is associated with destruction of all pathogens, but not spores. So in disinfection, all organisms may not be killed. All organisms may not be killed. But it is reduced to a number which is not harmful to humans. The surface has been disinfected. The number is reduced to a level. Number is reduced to a level that is non-harmful. It is reduced to a level that is non-harmful. This is called as disinfection. So in sterilization, the surface was totally sterile. Versus disinfection, where it is disinfected, it is reduced to a level that is not harmful. Next point. What do we mean by, what are antiseptics? Antiseptics, they are chemical disinfectants. Antiseptics, bache, they are chemical disinfectants. They are chemical disinfectants. That is called as antiseptics. Versus the next is asepsis, disinfectants. And the last basic term is asepsis. Asepsis, it is a technique which prevents the recurrence of infection. Asepsis, that means follow aseptic precautions. It is a technique by which occurrence of infection is prevented. It is a technique by which occurrence of infection is prevented. That is what we mean by the name follow aseptic techniques. That is, it is a technique by which occurrence of infection is prevented. These are the basic terms that we use: sterilization, disinfection, antiseptics, which are the chemical disinfectants or antiseptics, and asepsis. Asepsis are the aseptic techniques which are followed. This was basic. This is where we actually start with our chapter, that is methods of sterilization. This is where we actually start with our topic, that is methods of sterilization. First point, I can divide the methods of sterilization into two main categories, that is the physical versus the chemical methods. I can divide the methods of sterilization. I can divide them into two main categories: physical versus chemical methods. Starting with the physical methods of sterilization. Physical methods of sterilization, the first and the foremost, that is sunlight. Common sense, water, and sunlight. These are the three best sterilization methods. Sunlight. Heat. It can either be dry heat. It can either be dry versus moist heat. We'll be doing dry versus moist heat in extensive detail. Heat. Ozone. Filtration. Filtration, that is, we are filtering the contents. Filtration or radiation. That is either the use of ionizing or non-ionizing radiation. We'll do all the